Look down at the cotton shirt you might be wearing right now. Just, take a look at it.
Yeah. Or even picture the hard shell of a crab washed up on a beach.
Right. And then think about the simple, the white sugar cube you just stirred into your morning coffee. What if I told you that on a molecular level, they're practically the exact same thing?
It sounds impossible, I know.
It does, but welcome to the deep dive. You handed us this fascinating stack of translated biology and chemistry notes all about saccharides, which we commonly call carbohydrates, or, just sugars. And you challenged us to find the story hidden inside all this dense chemistry.
Which is quite a challenge, but a really fun one.
Oh, absolutely. So our mission today is to decode it for you. We're going to explore how a few simple atoms serve as everything from the invisible currency of human energy to the literal physical scaffolding of the natural world.
It really is a massive paradigm shift. Like when you stop viewing sugars purely as a flavor or a dietary indulgence, and you start recognizing them as the foundational building blocks of life on Earth.
Okay, let's unpack this, starting with the word itself. So saccharide comes from the Greek word saccharine, which literally just means sugar. But from a chemical standpoint, like when we strip away the food labels, what are we actually looking at here?
Well, at their core, saccharides are the most widespread organic substances in nature, and their construction is shockingly elegant. I mean, they are made of just 3 atoms: carbon, hydrogen, and oxygen. That is it.
Just 3.
Just 3. Chemically, they feature 1 carbonyl group and multiple hydroxyl groups.
Wait, let's translate that for a second. Carbonyl and hydroxyl groups, what does that actually look like if we could, zoom in on it?
Right, okay, so think of the carbon atoms forming a spine, like the trunk of a tree.
Okay, I'm picturing it.
And the oxygen and hydrogen atoms stick out from that spine like branches. And those specific branches are incredibly important because they're highly reactive.
Meaning they like to connect with things?
Exactly. They're exactly what allow these individual sugar molecules to so easily lock hands with other molecules, including water, or with each other. But the truly amazing part isn't just their shape, it's their origin. The ultimate source of all these sugars is plant photosynthesis.
Oh, the classic biology concept. Plants take water and carbon dioxide and using sunlight, they create glucose and oxygen.
Precisely. So when you consume a saccharide, you are fundamentally consuming captured solar energy.
That is wow.
Yeah, plants act as natural solar panels. They lock the sun's energy into chemical bonds. And it's not just about human food either.
Yeah.
Like, saccharides are used industrially in ways that might actually surprise you.
Like what?
Well, take agar, for example. It's a saccharide extracted from marine red algae. It provides the crucial gelatinous material used in microbiology to grow bacterial cultures in petri dishes. And it's also widely used in pharmacy.
Captured solar energy. I really love thinking about it that way. So let's look at the absolute base level. Before these molecules start locking hands to build trees or, algae, they operate as solo artists, the single units monosaccharides.
Right, and the absolute superstar of that group is glucose. Grape sugar.
Found in grapes, obviously, but also in honey. And crucially, it's flowing through our own blood right now.
It is so fundamentally necessary for human survival.
Yeah.
I mean, it's used in medicine for intravenous drips to give patients direct, immediate energy.
Yeah, and industrially to synthesize things like ethanol and even vitamin C, right? It's basically the base currency of the body's biological economy.
Currency is the perfect analogy. It's the immediate circulating asset that your cells spend to fuel their constant microscopic operations. Every breath, every heartbeat, it requires that glucose currency.
But then you have fructose, or fruit sugar, also found in honey and fruit, but it's actually 30% sweeter than standard beet sugar.
30% is a huge difference.
Right. But what's really interesting is its medical application. Because it is heavily used as a sweetener for diabetics, its presence in the blood doesn't force the pancreas to release insulin the way glucose does. So wait, if fructose is sweeter than regular sugar and doesn't trigger insulin spikes in the same way, is it basically a biological cheat code for a sweet tooth?
What's fascinating here is how subtle shifts in that basic atomic structure completely change human biology. Fructose and glucose are isomers.
Isomers, meaning
They have the exact same chemical formula, but the atoms are arranged in a slightly different shape. And because of that structural difference, the body's machinery has to process it differently.
Oh, I see.
Instead of circulating as immediate currency that triggers an insulin response, fructose is primarily metabolized in the liver. So while it sounds like a cheat code because it bypasses the pancreas, it's really just a a different, highly specific metabolic pathway.
That makes sense. And sugars aren't just providing the energy to run the body, they're actually written into the blueprint of the body itself, like ribose and deoxyribose, the literal D in DNA. Those are monosaccharides too.
Yes. They form the twisting structural ladder sides of the double helix. It is a profound realization. It's not just fuel, it is scaffolding. Without these simple single sugars, you don't have DNA or RNA, you just don't have genetics.
Okay, so if glucose is this invisible energy currency zipping through our blood or, sitting inside a piece of fruit, how do doctors or chemists actually know it's there? I mean, you can't just put blood under a standard microscope and see tiny sugar crystals floating around.
Right, you definitely can't. You need to force the sugar to reveal itself chemically. There's a classic test for this using copper sulfate, which historically was called blue vitriol. And you mix that with sodium hydroxide. You add this deep blue solution to your sample and gently heat it.
And what happens? I know the color changes, but what is actually going on inside the test tube?
What you're triggering is a chemical reduction. Because glucose is what we call a reducing sugar, it actively interacts with the copper ions. It's essentially a chemical robbery.
Robbery?
Yeah. The glucose molecule literally steals electrons from the copper. As the copper loses those electrons, its physical structure and how it reflects light completely changes. So as it heats up, you literally watch the blue solution shift to green, then to yellow, and finally it settles into a bright brick red precipitate.
Oh, wow. So it's not just a color change like mixing paint. You are watching energy and electrons being transferred in real time.
Exactly. It is chemists physically rendering that invisible currency visible. That brick red color is the definitive confirmation. It says, yes, there is usable biological energy in the sample.
That is so cool. So we have these single molecules stealing electrons and fueling cells. But what happens when these solo artists start combining? When molecules start mingling, I imagine biology gets complicated.
It definitely does. When two monosaccharides link together, they form a disaccharide. And the mechanism of how they join is beautifully simple. It's called a condensation reaction.
Condensation meaning water is involved.
Yes. When the two sugar molecules lock hands, they literally squeeze out and drop one molecule of water in the process.
And the most famous resulting duo has to be sucrose. Good old-fashioned beet or cane sugar. It's just a molecule of glucose and a molecule of fructose holding hands, minus one water molecule.
Which is found abundantly in nature. I mean, sugar beets can be up to 20% pure sucrose by weight.
20%. And human beings have done wild things with sucrose. Like, if you heat sucrose strongly, it undergoes a transformation and turns into caramel. Specifically, it becomes food additive E150.
Right, which is everywhere.
It is. And this isn't just the gooey stuff inside candy bars. They use this caramel to color dark beer, vinegar, and rum. We even engineer how we consume it. Did you know the actual physical sugar cube was patented in 1843?
1843.
By a man named Jacob Christoph Rad in the town of Dutchies.
That's a brilliant example of early food engineering. We took this natural, energy-dense molecule from a beet, figured out how to extract and crystallize it, and then standardize it into a perfect, uniform cube for parlors and tea times across the entire world.
But sucrose isn't the only duo. You've got lactose, or milk sugar, which is glucose paired with galactose. It's found in mammalian breast milk, and it's the sugar that ferments to create lactic acid.
Yeah, and then there's maltose, which is a powerhouse in human history. It's simply two glucose molecules locked together.
Now, I know maltose is absolutely huge in brewing beer, but where does it actually come from in the first place? Do brewers just buy bags of it?
No, actually. It comes from the biological breakdown of starch in germinating barley.
Oh, germinating like when it sprouts.
Exactly. When the barley seed starts to sprout, enzymes break down its massive starch reserves into smaller, two-part maltose molecules. Brewers then use hops and yeast to ferment that specific sugar.
So wait, let me get this straight. When I look at a pint of dark beer, I'm potentially seeing caramel coloring from heated sucrose. But the alcohol itself was made possible by the yeast fermenting maltose from the sprouting barley.
That's right.
Beer is essentially a masterclass in disaccharides.
It absolutely is. We haven't just passively consumed these chemical pairs. We've hijacked them to build entirely new food aesthetics, massive global industries, and ancient social rituals.
Man. So we've seen how pairing just two sugars changed human culture via brewing and tea. But what happens when biology needs to survive a harsh, sunless winter? Linking two sugars isn't going to cut it. Nature has to build massive biological storage vaults.
And that takes us into the realm of the polysaccharides. We are no longer talking about single or double molecules.
We're talking big chains.
Huge chains. We're talking about linking up to 30,000 glucose molecules together. These are heavy-duty reserves, and nature has developed two distinct strategies for managing these vaults, one for plants and one for animals.
Right, so for plants, the storage vault is called starch. Its Latin name is amylum, and it's heavily concentrated in tubers and seeds. Think potatoes, wheat, rice, corn.
Right.
What's strange is its physical property, though. It's a white powder that is completely insoluble in cold water. It just sits there. But if you put it in hot water, those grains suddenly swell up into a thick, viscous paste.
Which explains why the modern food industry is entirely dependent on it.
Right. It's used industrially for glue, for stiffening textiles, and paper manufacturing. And it is the hidden thickener in ketchup, yogurt, and salad dressings. The ketchup in my fridge is thick because it's packed with microscopic plant energy vaults.
It's true. And just like the copper test for glucose, you can physically see these vaults if you know how to look.
Really?
If you take a scraped potato and drop a simple iodine solution onto it under a microscope, the iodine reacts with the coiled structure of the starch. The grains turn a deep, distinct blue.
Oh, that's fascinating. But humans and animals don't store starch. Like if I eat a potato, I don't turn it into plant starch in my body.
No, we don't. We store glycogen. You can think of it as animal starch. It's stored primarily in our liver, which can actually be up to 20% glycogen by weight.
20%.
Yeah, and in our skeletal muscles, which hold about 7%, it sits there, highly condensed, and breaks back down into single glucose molecules during starvation or intense physical exertion.
Okay, here's where it gets really interesting. It's like our body's internal financial system.
Oh, I like that.
Yeah. Your muscles are the checking account. The funds are right there, locally stored, ready for quick, active use the second you need to sprint away from danger or lift something heavy. But the liver, that's your deep savings account. It regulates the whole system, slowly releasing glucose back into the blood to keep the lights on and keep your brain functioning when you haven't eaten in days.
If we connect this to the bigger picture, you really see the stark evolutionary contrast between the kingdoms of life. Plants are stationary. They have the luxury of hoarding massive amounts of dense, heavy energy as starch in a root or a seed, just waiting for the next season.
Because they don't have to carry it around.
Exactly. But animals, we are mobile. We run, we hunt, we hide. We need an energy storage system that is lightweight, highly branched, and capable of being mobilized back into blood sugar in an absolute instant. Glycogen gives us that immediate, on-demand liquidity.
We need energy on the go, and glycogen provides it. Now, we've covered how sugars power life and how they store life's energy, but there's a huge leap from storing energy inside a cell to physically building the world around us. These megachains aren't meant to be broken down for fuel. They are the architectural scaffolding of nature itself.
And this is where we meet the absolute heavyweight champion of organic chemistry, cellulose. It is the most widespread organic compound in nature, period.
Wow.
Is the main building block of plant cell walls. Wood, cotton, hemp, flax, it's all cellulose.
And we use it to make paper, cellophane, cotton wool. But here's the part that always trips me up. If cellulose is just a massive chain of glucose molecules, why can't we digest it? I mean, we eat it. It's what we call dietary fiber, which is vital for keeping our intestines healthy and moving, but we get zero energy from it. Why can we digest the glucose chain in a potato, but not the glucose chain in a stalk of celery?
It all comes down to the microscopic geometry of the chemical bonds. The single difference is the exact angle of how those glucose molecules link together.
The angle.
Yeah. In starch, the bonds are all oriented the same way, creating a spiral shape that our digestive enzymes can easily snip apart. In cellulose, every alternating glucose molecule is flipped upside down.
Oh, imagine it like a zipper on a jacket.
Okay.
Starch is a normal zipper. Our enzymes are the zipper pull. They just slide down and easily separate the teeth for quick energy. But with cellulose, nature flipped every alternating tooth upside down. It forms a straight, extremely rigid cable, and our human zipper pull just gets jammed. We simply can't break it.
That is a perfect analogy. The human zipper pull gets completely jammed.
Mm.
But certain animals, cows, termites, horses, they can digest it.
Wait, do they have a better zipper pull?
Not exactly. They've essentially outsourced the job. They have symbiotic bacteria living in their digestive tracts that possess the exact right chemical key to unzip those flipped bonds.
Wait, pause for a second. Think about what we've covered today. The paper we're reading right now, the cotton t-shirt I'm wearing, the dietary fiber in my salad, and the sugar cube I put in my coffee. They're all built from the exact same atomic ingredients.
Yes, carbon, hydrogen, oxygen. And cellulose isn't the only structural builder, by the way. There's inulin, which is found in dandelions and chicory root. It's actually harvested and used as a pastry additive for diabetics because of how it behaves in the body. And then there's chitin.
The crab shell.
Exactly. Chitin forms the cell walls of fungi, like mushrooms, but it also forms the hard exoskeletons of insects and crustaceans.
That is wild. My cotton shirt, a mushroom, and a crab shell are all utilizing the same fundamental chemical family to build their structure. And speaking of that structure, there is a very practical environmental note you brought to our attention from the sources.
Yes.
Because cellulose is such a robust, rigid cable, recycling paper is vastly cheaper and significantly kinder to the environment than constantly harvesting new wood to extract it. We're basically just reusing nature's scaffolding.
Right, and this raises an important question about the profound ecological implications of these structures. Think about how the simple inability of humans to break down that specific cellulose zipper completely shaped our evolution.
How so?
Well, because we couldn't just eat grass or tree leaves for energy. We had to become foragers of fruits, hunters of animals, and eventually inventors of agriculture to cultivate easily digestible starch like wheat and potatoes. Meanwhile, animals that formed those symbiotic partnerships with bacteria unlocked access to the most abundant source of energy on the planet, plant stalks and leaves.
It is truly an incredible journey when you step back and look at it. I mean, we started with single molecules captured drops of solar energy zipping through our blood as an invisible currency. Then we watched them pair up to sweeten our tea and brew our beer. We saw them bundle into chains of 30,000 to thicken our ketchup and act as our liver's emergency savings account.
It's a lot of ground to cover.
It is. And finally, we saw them weave into massive rigid cables to build the trees, our clothes, and even the shells of crabs. Next time you put on a cotton shirt, or notice how thick your ketchup is, or stir a sugar cube into your cup, you'll know exactly what invisible chains of carbon, hydrogen, and oxygen are making it all possible.
It really forces you to respect the elegance of organic chemistry.
So what does this all mean? Where does this leave us at the end of the day?
I think it leaves us with a truly mind-bending hypothetical to ponder. As we discussed, our bodies run exclusively on glucose, but we lack the enzymes in the symbiotic bacteria to digest cellulose, which is nothing more than a massive locked-up chain of that exact same glucose.
Right.
I want you to imagine how human history, how civilization, and how the entire modern food industry would have been radically altered if we could break it down. If we had that one microscopic bacterial partnership in our gut, we could literally eat grass or paper or twigs for energy. Starvation would be practically impossible in a forest. I invite you to ponder how the absence of one tiny bacterial partnership changed our entire evolutionary trajectory and our complete relationship with the planet.
We'd be a completely different species, and it all comes back to a tiny microscopic shape. Is it a sugar you can burn, or is it a sugar that builds a tree? It's the same atoms, just a different design. Keep that in mind the next time you look at a spoonful of sugar.